It gives me great pleasure to introduce our next speaker, Ranjan Sen, who's the Chief of Laboratory in Molecular Biology and Immunology at the National Institute on Aging.
Okay, thank you very much, Steve, for the introduction. And a big shout out to the organizers of this terrific meeting. The community, which has been wonderful, is I was just saying, a closely guarded secret. I feel I've been working on antibody genes for a while and only just realizing that you guys exist. So that the next step when I get back will be to sign up and become a member of your community. So that's a promise, and so we'll go from there. So my talk, and I told Stephen to feel free to haul me off the stage if I'm going longer than I'm supposed to, is going to be about generating the repertoire and genetic and epigenetic control of antigen receptor gene assembly. So let's see, let me see if I can work this. Okay, so my topic today is the mouse immunoglobulin heavy chain gene locus. And as everyone in this audience knows, the locus is encoded in the genome as a series of gene segments, variable gene segments, diversity genes, variable gene segments, diversity gene segments, and joining gene segments. And during B cell development in the bone marrow, one variable gene segment, one diversity, and one joining gene segment is put together by the forces of VDJ recombination that was just talked about in the previous talk to generate the exons that encode the variable part of IgH chains. But this simplistic textbook version of what the locus looks like can be elaborated when we look at the scale schematic of the IgH locus in mouse, which encompasses about 3 megabases of the genome, the majority of which encodes these variable gene segments marked in different colors by the different gene families by their old nomenclature. Only the 3' end of this locus, about 300 kilobases of it, encodes the diversity joining gene segments and the exons that encode the heavy chain isotypes. So at the bottom part of the slide here is our own analysis of the pro-B cell repertoire isolated from bone marrow of mice. And the striking feature of this is something that I think that this receptor repertoire community already knows, that essentially all these variable gene segments that are spread over such a large expanse of the genome are used at approximately comparable efficiencies. Now, sure, you can see ups and downs, but in general, most of these V gene segments, there are 100 of them, functional ones in the mouse, they're all used in the pre-immune repertoire as it is generated in the bone marrow. So the question is, what are the forces that allow these gene segments that are spread out across the genome to come together, to be put together during VDJ recombination to encode heavy chain genes? So prior to the initiation of recombination, there are 3 regulatory sequences that I will be dealing with. That's the genetics part of my talk, not the kind of genetics we've been hearing about. And those 3 cis-acting regulatory sequences are labeled here as IGCR1, intergenic control region 1, that's the green oval. The enhancer that is located between the joining gene segments and the constant region exons, referred to as EMU. And then a cluster of DNA's hypersensitive sites are the 3' end of the genome that are also referred to as the 3' regulatory region or the 3' CBEs, where CBE stands for CTCF binding elements. Okay. So these 3 regulatory sequences coordinate in a way in order to ensure generation of the repertoire. The first step of which is, of course, the introduction of one diversity and one joining gene segment to make, this DJ recombined allele. DH recombination is regulated in at least 3 ways that we know of that are listed here. D gene recombination occurs before V gene recombination. The 5' most and the 3' most D gene segments are used most frequently, and they are the gene segments in mouse labeled here as DFL16.1 and DQ52. And a mechanistically incredibly interesting part of D gene recombination is the fact that D gene recombination occurs by both deletion and inversion. And that's because D gene segments are flanked by recombination signal sequences that the VDJ recombinase recognizes. So in principle, they could recombine by deletion or inversion, but the preference for deletion is about 1,000-fold. Okay? Now, we will not further discuss this part, but if anyone is interested in those mechanisms, we can talk about it after the talk. So we, several years ago, showed that the enhancer, the emu enhancer located over there, configures what I will refer to as a 3-loop pre-rearrangement structure of the IGH locus. And that's because the enhancer interacts with the intergenic control region 1, which is about 70 kilobases away. It also interacts with the 3' CBEs located about 200 kilobases away, and an inferred small loop in which the enhancer talks to the promoter associated with the 3' most D gene segment. We imagine that this looks something like this, which is why I call it a 3-loop configuration. But I caution you that we, in fact, do not know how many cells, how many developing pro-B cells actually have a 3-loop configuration. It is very likely that it's a distribution of configurations where either you have interactions between only 2, either here and here, or there and there, and etc. So it's a whole mix of things. And in fact, I've been saying this ever since we proposed this configuration, but only about 2 or 3 weeks ago in one of the latest issues of Science, where people have actually measured the amount of time that putative loops stay together. And this was measured not in the heavy chain locus, but in embryonic stem cells using some other gene that has been proposed to form associations between 2 sites 500 kilobases apart. And they found that only about 6% of the cells actually have a closed configuration. So I belabor that point so that even though I talk about loops and interactions and associations, you should not take away the picture that this is sealed in cement in every pro-B cell. Okay, these are sites of preferred interactions between these cis-regulatory sequences, and their identification allows us plausible mechanistic explanations for many of the features of VDJ recombination. And at the moment, we are dealing with the first step of VDJ recombination, D to J. So proposal of the structure has some interesting implications regarding those 3 properties of D to J recombination. First of all, we have this small or this intermediate loop of about 70 kilobases. This encompasses all the diversity gene segments and excludes all the variable gene segments which lie in the genome way away from here. And the first V gene segment is about 100 kilobases away from the base of that loop. And we propose that the fact that these D gene segments are all together was one of the reasons that they recombine first. This goes along with the determination that this small green loop, the inferred loop between the enhancer and the promoter of DQ52, is also the place where RAG recombinase has the highest levels of binding in the heavy chain locus prior to the initiation of recombination. Okay, this is referred to as the recombination center, and the fact that these diversity gene segments lie close to the recombination center, whereas the variable gene segments lie far, far away, may be one reason why D genes recombine first. Additionally, the 3' most D gene segment and the 5' most D gene segment are the ones located spatially most closest to the recombination center. And we propose that that might be one of the reasons that the 5' most and the 3' most D gene segments recombine at higher frequencies than the intervening D gene segment, which will lie in the rest of the red loop there. Okay, so with the relative explanatory potential of this configuration, our next question was, how does the enhancer configure this structure to the locus prior to the initiation of recombination? And so we've used tools of genetic manipulation to perturb the locus and then to reassess what is the state of this spatial configuration, and to assay the effects on recombination and gene expression transcription. And I'm going to show you just some examples of the kinds of data we get in this slide and in the next couple. The reason that this becomes even more potentially interesting as to how does the enhancer configure this structure is because these two regulatory sequences, the IGCR1 and the 3' CBEs, are both binding sites for this transcription factor called CDCF. But the enhancer, which we think orchestrates this configuration, does not bind CDCF. So we explored these possibilities using, nowadays, using these technologies, which are developments of the chromosome conformation capture technology developed by Job Necker. This is shown here is a high C, a capture high C. A high C looks at every interaction in the genome, but since we are only interested in the heavy chain locus, we pull out of that high C interactome only the ones that refer to the heavy chain locus. And the way, and if you just focus on the left part of the screen here for the moment, the way this data is interpreted is to see that these dots, these spots that have higher density compared to the rest of the background represent interactions. So for example, this dot here represents the interaction of the enhancer with the 3' CBEs. This one represents the interaction of the IGCR1 with the 3' CBEs. And this little dot here represents the interaction of the enhancer with the IGCR1. Okay. I'm not going to I'm not going to show you too many of these graphs, but I'd just like you to get an idea of the kind of analysis that is going into the statements that I'm going to make. And this, by definition, is a population analysis. So we are not looking at it in a single cell. And I think that has its associated caveats that we can discuss. On the right-hand side is a so-called high chip analysis where we ask which DNA sequences are interacting with each other when we pull down, immunoprecipitate a variety of different transcription factors. In this case, the high C map is done with RAD21, which is a component of the cohesin complex, and the cohesin complex has been shown to interact with the transcription factor CDCF. We do the same kinds of experiments by pulling down CDCF, or we can do the experiment of pulling down other histone modifications to ask how specific sites of histone modifications interact with each other in the genome. But when you look at this map, you also see essentially the same things. You see the same idea, the interaction of the enhancer with the 3' CBEs, the interaction of the enhancer with IGCR, and so forth. So what happens when you remove the enhancer from this configuration? And that's shown on these contact maps. I'm showing you only the high chip analysis was RAD21, and the simple result is that any enhancer-associated interaction, such as that one with the IGCR, disappears, or that one with the 3' CBEs disappears. Okay, so no enhancer, no interaction with the enhancer, as you might expect, but no other change in the rest of the interaction. The IGCR continues to interact with the IGCR continues to interact with the 3' CBEs over here, and it also makes some contacts with the proximal variable genes, which we will discuss in just a couple of slides. So conversely to what happens when you remove the enhancer, where nothing else changes other than the enhancer-dependent interactions go away, we think one interpretation of this is that the enhancer blocks cohesin-associated generation of these loops. But if you do the other experiment where you delete the intergenic control region, now you see new other things going on. So in particular, the ones that I've marked in red, these are loops between the enhancer and IGCR1. They go away. These are loops between the IGCR1 and the proximal Vs. They go away. But instead, the enhancer now begins to interact with the proximal Vs and so forth. So what has happened is that one has altered the configuration from interactions of the enhancer with the IGCR with these regions to the enhancer, which is one of the reasons that I started by saying that the enhancer nucleates or drives the configuration of the locus. So what are the consequences of doing this? So one of the consequences is that this larger interaction of the enhancer on IGCR deleted alleles now incorporates a D gene segment that is normally very infrequently used in VDJ recombination. And that's this element known as DST4.2, which is located about 60 kilobases away from DFL16, which is normally the D gene segment that is used most frequently. So this D gene segment now gets used, and we imagine the new configuration to look like this, where this intermediate 70 kb loop is now increased to about 170 kb loop. The other parts have remained unchanged. This new loop now, DFL16 moves away from being near the recombination center, and a new DST4.2 is brought into this loop. The consequences on VDJ recombination can be assayed by using specific primers that recognize either the different D gene segments shown here in different colors, these triangles here, and a primer that recognizes the joining gene segments. And so depending on which combination you use, you can assay which D gene segments are represented in the genomic DNA that you're looking at. So the data I'm going to show you comes out of pro-B cells obtained from genetically altered animals. So what you can see is that if you use DFL16 as the primer pair, compared to wild type on IGCR deleted alleles, the frequency of DFL16 recombination goes down. The frequency of DSP2s, the intervening D gene segments, does not change. And when you look at the newly incorporated DST4.2 that did not recombine on wild type alleles, now that begins to recombine at reasonable levels on IGCR deleted alleles. Okay? So in these altered structure that is generated when you delete the IGCR and the enhancer now interacts with a more distant site, the consequence is that DFL16, which moves away from the recombination center, its recombination frequency goes down. DST4.2 that moves into this newly generated chromatin domain, its recombination frequency goes up. And so our takeaway from that is that DH utilization and frequency is determined by the pre-rearrangement locus structure. And that pre-rearrangement locus structure is nucleated by what sequences the enhancer interacts with. Does it interact with IGCR1 on normal alleles or with other sites, which I'm going to talk about, on IGCR deleted alleles? The second step of heavy chain recombination is, as you all know, the recombination of one of these variable region gene segments to the DJ junction. Okay, and this, I think one of the striking features of V recombination not only is the fact that there are these many V gene segments. Okay, I've got my hint, so I will, if you find me accelerating, that's only because my time's running out. But okay, so not only is the fact that all these spread out V gene segments can recombine in, but they do precisely to the DJ junction and not to the unrearranged D gene segments that lie upstream of the DJ junction. Okay, because depending on which D gene is used to create the DJ junction, you could have unrearranged Ds. Those in the mouse are located within 4 kilobases of the DJ junction. Yet when V's recombine from a megabase away, they find the DJ junction, but not the 4 kb upstream other D gene segment. I think that's, I mean, to me, that's a remarkable example of specificity of the recombination reaction. And in a paper several years ago, we proposed that DJ junction is marked distinctly from the other D genes. But this order of recombination, Ds first and Vs, breaks down on IGCR deleted alleles, in which Fred Olt's lab several years ago showed that the VH can now recombine to an unrearranged D gene segment. Okay, so how does this come about? And I will take just a few minutes to go through that and conclude. But to sort of explain what we think is going on, I need to give you a slightly blown up version of the proximal V genes, which is shown down here. So far, we have been discussing the diversity and joining gene segments over here. The functional V genes are shown here in the solid boxes. The hatched, with the broken line, those are pseudo V genes. And the diamonds here represent CTCF binding sites. So what we found using these chromosome conformation assays is that on IGCR deleted alleles, the enhancer, instead of making this 70 kb loop, makes this 170 kb loop that goes precisely to the first available CTCF bound site in the genome. In doing so, it incorporates, of course, the DST4.2 gene segment that we've been talking about, and now has places a variable gene segment close to the recombination center, because this is now the big loop. Okay, and there's one additional feature of that gene segment going there that I want to quickly go through in the next couple of slides, and that is that V gene segment now undergoes high levels of inversional recombination, which has never been observed on the heavy chain locus, in part because the V genes recombine to DJ genes. Okay, but here they undergo inversional recombination. I will spare you the actual experiment that show that, but it's again using the right combinations of primer pairs to be able to detect this kind of inversion. And you can detect inversion by looking at the VD junction itself, or by looking at this so-called signal sequence junction that forms, which is shown on the slide here. So taken together, what the one message that I'd like to leave you with is that distinct rules of engagement operate on DH and VH rearrangements. And I won't go through this schematic in detail because for time's sake, and I'm open to questions here, but you should realize that the rules by which recombinase recognizes the same, essentially, very, very similar recombination signal sequences, be they on D genes or V genes, is different. And the reason it does it is because of the structure of the locus that permits recombinase certain RSSs in certain orientations. So let me just skip over a few slides here to leave you with my other major conclusions. So regulation of IGH rearrangements is what we've been talking about. DH rearrangements occur within a domain. DST4.2 being brought in leads to its recombination. They do not require an associated CTCF binding site. And spatial proximity to the recombination center increases frequency. VH rearrangements, on the other hand, occur from outside an EMU configured domain. They do require an associated CTCF binding site. And again, if you are proximal to the recombination center, you get more efficient recombination. Okay? So what does it mean for the entire locus? We think that the proximal genes in the mouse locus, at least, are depend upon the creation of this smaller loop when emu does not interact with IGCR, shown in the dotted lines, and that the long distance interactions that allow distal V recombinations are blocked on alleles that emu and IGCR interact, which together form a strong stop for quote unquote loop extrusion that leads to the bringing of V genes close to the recombination center where they will undergo recombination. So I'll stop there and take any questions, and I'm just trying to stick to time.
So there are nuclear matrix attachment regions that flank the heavy chain enhancer, and I'm wondering if attachment to the nuclear matrix is playing any role in organizing that recombination center.
Right. So you're absolutely right. First of all, nuclear matrix attachment regions were identified by Bill Gerard and Peter Cockrell many, many years ago. So there's two things I can say about the MARS, the matrix attachment regions. When they have been looked at in transgenic mice, now that's different from the endogenous locus, but when you make transgenic mice and you make it with or without MARS, those were experiments done by Rudy Großschädel many years ago, they don't seem to make a difference in expression of the transgene. Now, those expression of transgenes did not require recombination. They were pre-recombined, VDJ recombined genes. So the question is open whether one actually makes deletions, selective deletions of the MARS, will they affect recombination or recombination center formation? So that's not known. But conversely, if you leave the MARS there and selectively delete the enhancer, which we have done, that will abolish recombination, or at least substantially diminish VDJ recombination and alter the structure. So for example, the structures that I showed you. So the MARS are not sufficient in the endogenous locus. Whether they are necessary is not known.
Fantastic talk. A question to the rules of DH and VH. Do you have a notion of how they maybe vary across the population? Are they more similar for twins than for non-twins, for example? Has this been looked at? Yeah.
Right. So at the outset, my analysis so far is restricted to mice.
Yeah.
So I have no idea how they may or may not change in the context of human genetic variation or not. My bias is that if the structure is maintained, then the phenotype will follow. Whichever Ds are used or not used. But it opens. But this analysis, from the perspective of what most of you are interested in, I think raises the question whether SNPs or polymorphisms in those sites that direct the structure, they will change D or V usage, even if the actual sequence of the V gene or the D gene segments are the same. So at our institute, we've just got an IRB for human bone marrow. So we'll be trying to do some of these, recapitulate, or at least see to what extent the studies in mice recapitulate in human pro-B cells in terms of both structure and the epigenetics. But I have no answers there yet.
This may be naive, but what part of this would be epigenetic, or how would you? Why are some of this epigenetic and some of this is genetic?
Well, so the way I think about it, that you need the genetics in order to have the locus do, you need an enhancer to activate transcription. You need an enhancer to make loops. Yeah. But without making the loops, which is the epigenetic part, because that is not direct, that is not changing the sequence, but it's taking the altered sequence and altering the structure. Without the epigenetic part, the genetic readout would not be possible. So the epigenetics, I think, simply reads out the genetic code that is in the sequence per se.